Hi, my name is Kishan Nandakumar, and today we will be comparing the fungus borrowed from ancient bacteria to defy gravity and see if it accurately describes the research article Evolutionary Novelty in Gravity Sensing Through Horizontal Gene Transfer and High Order Protein Assembly. Some important Bio 282 topics to know for this presentation is that mutations arise in the DNA randomly so that they don't purposefully benefit or harm the organism's success. But these beneficial mutations can lead to the evolution of organisms, as these genes will be naturally selected for and they'll get passed down over many generations. Another important thing to know is that there are two main protein structures that will be covered in this research, which is tertiary and quaternary. And the function of these proteins is based on their structure and interaction either between the amino acids or with other proteins. So this news article goes over a fungus called the Phycomyces blakesleanis, which was studied to figure out how these kind of fungi can actually grow toward light sources and also sense objects around them. And this article goes over a study by Gregory Jed that actually shows that these fungi have gravity crystals, which mean that they have evolved from past bacteria, which might have actually given these gravity crystals to the fungi through a process called horizontal gene transfer. Once this gene was given, it probably led to a beneficial mutation that allowed these fungi to have higher rates of success for their spores due to the use of these gravity crystals. Okay, now we will be kind of discussing the research article, which is the evolutionary novelty in gravity sensing through horizontal gene transfer and high order protein assembly, which was completed by Gregory Jed and his colleagues. His colleagues are Tu An Nguyen, Jamie Greig, Asif Khan, and Kara Go. Okay, so for this article, we'll be discussing some of the methods that were used. So first, these researchers grew the Phycomyces blakesleanis fungi, and then they were able to actually identify 3 different peptides. And when they ran these 3 different peptides, they called them the octin gene, and a BLAST-P analysis was run on them. And using this BLAST-P analysis, they actually constructed a species phylogenetic tree, which, as you can see on the right, it's not the exact species, it's just kind of made up species, but this is kind of what the phylogenetic tree shows, and it shows that one ancestor would lead to the next ancestor, and it kind of shows how these fungi evolved these crystals from the ancient bacteria. Once this species tree was created, these fungi crystals were then disassembled and their protein structure was further studied. Once these methods were done, the results showed that a mass spectrometry showed that the protein was actually octin, which we kind of discussed in the methods. The BLAST-P analysis revealed that this octin protein was actually present in primitive forms, but it was present all the way back to proteobacteria. Once these crystals were disassembled, it was found that actually disulfide bonds and non-covalent interactions stabilized the tertiary protein structure, which, as you can see on the right, this protein structure uses these disulfide bonds within the cysteine amino acids to be held together. More analysis of protein structure also found that there may have been a mutation that actually gave the fungi an advantage when it came to the horizontal gene transfer from the ancient bacteria to the fungi. So as you can see, this is a picture of the results, and this is actually measuring the time in hours for the Phycomyces fungi to grow, and the asterisk with a bracket next to it is where the octin protein codes for the crystal, where these gravity sensing crystals are found. And the picture underneath is actually the close-up of these gravity sensing crystals. So one of the main things is you can see that these gravity sensing crystals are clustered together, which allows them to actually increase their size, meaning that they're able to find the nutrients more effectively and find the sunlight more effectively, which is important in the self-assembly of these crystals because the self-assembly of these crystals actually makes these crystals much more effective for the fungi. So after reviewing the summary of the news article and the methods and results of the research, does the news article accurately represent the research article? And to this, the answer would be no, it does not. Okay, so why does the news article not accurately display the research article? Well, for one, although the article does report on the findings of the research, it leaves out many integral parts of the research that would likely be very necessary for the readers. The article does fail to mention the importance of the phylogenetic tree, which, as we showed a picture of before, was probably one of the most important things in determining that these proteobacteria actually had these gravity-sensing crystals before the fungi did. Also, the article leaves out the important structures, as we also showed in the picture, that are involved in the protein creation, such as these disulfide bonds and these non-covalent interactions, which would be extremely important to know both the structure and the function of these proteins. The article is also incorrect in one way because it reports conclusively that these proteins are self-assembling, which form the crystal, when in reality, the experiment actually indicates that there should be more research done to find out the true control of these proteins. All in all, the news article does not really accurately display the research article, and if people do want to find out information about topics like these, it is much more advisable to actually read the research and at least try to get a summary of it for themselves rather than just trying to take a shortcut and reading the news.